Hello, I am Minkyu Park, and I founded Eucrogen. First of all, thank you for giving me this very interesting opportunity to present about my research. And this study is completed when I was working at National Cancer Institute. So this is about the precision oncology to utilize the AI model. The title is WSIomics, an end-to-end pipeline, end-to-end multimodal deep learning pipeline for binary classification using whole slide images and transcriptome datasets. In this case, the whole slide images means the microscope image of the cancer tissue, and transcriptome dataset means I use the gene expression information and microRNA expression datasets. So yes, this is about the precision oncology. Precision oncology is the cancer treatment method for the personalized treatment. So before, usually biomarkers were used a lot. So using the biomarkers, scientists who tried to predict the prognostic of the cancer. For example, using the biomarkers, the risk of the cancer patient was stratified. For example, using the biomarkers, cancer patients was classified into low risk, intermediate risk, and high risk groups. For example, in the case of high risk, the cancer patients can die earlier. And if the cancer patient is classified as low risk, then it means they can survive longer. So using the biomarkers, this could be predicted. Also, the other part was the classification of the therapy response. Using the biomarkers, the scientists could predict if the treatment will work well or not to the cancer patient. So before the biomarkers were developed and tried to use to this precision oncology. But as more clinical data sets are accumulated and the AI technology advances, people try to improve the precision oncology method using the multimodal AI model. So now it is still early stage in this AI-based precision oncology, but it is very fast-growing research area. So this shows the data set used for multimodal AI model to develop for the precision oncology. There are four representative data modality. One is the clinical data set. The clinical data set include cancer stage, tumor types, age, gender, and there are a lot more clinical data type. And also, radiological data sets such as CT image is also one of the popular input data. And the most popular data is pathological data. As I told you, the whole slide images, which is H&E stained microscope image of the tumor tissues. Through this, the microenvironment of the cancer tissue uh, are investigated, and this provide a lot of information. And also, uh, genomic information such as SNV, methylation, and transcriptomic profiling data can be used. So these can be, by combination of these data set, the multimodal AI model can be trained. And this shows how the multimodal data set can be fused to train the multimodal AI model. There are multiple methods, but largely it can be classified into early fusion, late fusion, and intermediate fusion. Early fusion is the merge the features or raw data from the beginning. So, for example, if the input data is a numerical data like gene expression value or patient's age, then we can just fuse them in the early stage by after scaling. So that is the early fusion case. And in the case of late fusion, um, that is done by training individual model in each modalities. And if it predicts the final classification, then using the final results, it just predicts the result again. The simple example is like, for example, there are three modality and two predict it is A, and if one predict it is B, then because two predicted it as A, then we can choose it like A. That is the one simple example of the late fusion. And the third is intermediate fusion. It is the mix of the early fusion and late fusion. In the case of intermediate fusion, it train uh, each of the model in each of the modalities, and it merged not the predicted result, but in the case of deep learning, there are multiple layers of the parameters, and it just merged the final layer, the parameters in the final layers, and using that merged parameters, it trained another model that is for the multimodal model. So so if we use two modalities, then it will train three AI model. One is two are from each modality, and the third will be the multimodal model. So in my study, I use this intermediate fusion method. So in my study, I used the whole slide image and transcriptome profiling data, and this shows the one example of the whole slide images. So this is a thin sliced tumor tissue and cancer tissue, and it is stained with HN stained. This picture, the original picture of the horse slide image is very big. Usually, if we take a snapshot in our daily life, uh, the picture file size will be around 1 MB or 4 MB. But in this case, the file size is 1 TB. So if we enlarge this picture, then we can see the cell structure like nuclear and cell boundaries. So this is extremely high resolution data and very big size picture. So this is a popular material to investigate about the cancer. So based on this, scientists can predict the survival of the cancer patient, like the patient will live longer or shorter. And also based on this, the scientists can predict the therapy response, like if this patient will respond to this drug or the treatment by this drug or not. And also, I saw some cases, some research who predict gene expression, which is related to cancer trait. Based on these images, um, the AI model can predict expression of certain genes that are related to the cancer. So I can say this contains a lot of information to determine the cancer-related clinical data. So in my case, I utilized this whole slide image to predict the therapy response by along with the transcriptome data. So there are several software packages that can train AI model with these whole slide images. Um, I could see the Harvard University and Stanford University are leading this part to process these whole slide images. In my case, I used the software package developed by Harvard Medical School, and the software package name is CLAM, and it used attention algorithm. And the reason why I chose this software, this tool is, in the case of attention algorithm, um, it is more powerful with a smaller number of the input samples. Uh, in other case, other software use the transformer algorithm. It is actually more powerful, but to utilize the transformer algorithm, more data sets are needed. But in my case, I used not enough data set, so this attention algorithm was worked much better. So I needed the data set to train multimodal AI model. So in the case of cancer research, uh, TCG database is one of the most popular cancer database. So uh, it stores a lot of various um, cancer-related data set database. For example, whole slide images and genomics data, clinical data, and also the transcriptome data are stored in this database, and people can easily utilize those for their research. But one problem was that this database was not constructed to train AI model because this database has very long history. So some data set are very old, and the data set are also collected from multiple facilities. So it is not well unified. So the data quality or quantity is not enough to train generalizable deep learning model. So I can say, um, because I use this database, the size of the data was not enough to train generalizable AI model. So this shows the goal of this study. The goal of this study is the development of a highly reproducible training pipeline for iterative training of the multimodal AI models. The reason why I tried to develop highly reproducible training pipeline is the not enough data sets. Actually, this is common to all the scientists who study about um, that this precision oncology. So there are so many studies who tried to develop high performance AI model to predict the prognosis of the cancer patient, but because of the data issue, none of generally applicable uh, model is developed. So what I thought is that initially maybe I can train initial model with this sparse data using the um, using the this pipeline, then we can easily expect the new data will be added in the future to this database. Then if new data comes, you can evaluate the performance of the existing model and report it to the users and retrain the existing model with this new data. Then the performance of the existing model will be improved and the new data will be saved into in this database. And and there will be another new data. Then we can repeat this process, like evaluate the existing model again. Then we can see some increased performance of the model and report it to the users. And retrain the model, existing model, then the model will be better, more improved. Then the users will see the performance changes. So it will be increased more and more as more new data is are added. Then if the performance reaches a certain degree, then the users can decide to use the real world data with the patient. Then they can utilize this AI model for the precision oncology. So this is the basic concept to develop this reproducible pipeline. So I try to develop a generally applicable training pipeline, which is applicable to various cancer types, and also it should be applicable to sparse data set because the current data sets are in sparse data sets. And also, to train the therapy response model, I needed the resource for clinical data of this TCGA pan cancer. In the case of TCGA database, they just provide the some simple clinical data, whole slide images, genomics data, and transcriptome data. So to determine the therapy response, I needed some additional information, and I could find it from this paper. So this paper provides the progression-free interval. So based on this progress-free interval, um, the therapy response can be determined. For example, progress-free interval is the time between initial treatment and cancer recurrence. So from the initial treatment, if cancer recurrence occurs very long time, and if it takes very long time until cancer recurrence, then your the doctors determine, oh, the treatment is working well, so the patient is sensitive to the treatment. But right after the cancer treatment, if the cancer recurrence occurs, then the doctors think, oh, this treatment does not work, so the patient is resistant to the cancer treatment. So if this progression-free interval is under 6 months, then the patient is determined as treatment resistant. But if it is over 6 months, then usually, for example, in the case of ovarian cancer, it is determined as treatment sensitive. But it is not unified standard or criterion across all kind of cancer. So it is very hard to find unify this criterion. So in my case, I just unified the uh, criterion of the to determine the resistant and sensitive. So in my study, if the PFI value is under 6, I classified it as resistant. And if the PFI value is over 36 months, then I classified it as a sensitive case. The reason why I did it like this is the purpose of this project was not to train generally applicable the AI model, but just to develop the pipeline that can be used for various cancer types. So to develop that pipeline, I used these two extreme cases. So actually, there were a total of 11,160 cases for 33 type of cancer in the database. But in this study, I used 10 cancer types. So this shows the 10 cancer types, and the numbers in the pink boxes shows the number of the patient case in um, resistant and sensitive patient case. So y-axis represent the PFI value, and x-axis is the sorted patient case by the PFI value. So, but as you can see here, the number of the samples between resistant and sensitive is different. So this can cause a problem when we train the AI model because, for example, in this case, there are much more, over 4 times more data in these sensitive cases. If there are more data set in one side, then when we train the AI model is more well trained with the sensitive cases. So there will be a severe bias towards sensitive cases. We call it overfit. So to prevent this overfit, um, I just balance the samples between resistant and sensitive. So in this case, with the smaller number, in this case, I used a 95 resistant sample, and among these 422 sensitive sample, I randomly selected 95 sample to make the equal number of the sample between this resistant and sensitive. That is commonly applied to all these 10 cancer types. And this shows the number of the used data sets. Um, The datasets were curated for testing the pipeline with therapy response across the 10 cancer types. So the columns represent the used cancer type, and rows shows the data modality. It is very complex, but you can just focus on the numbers highlighted with this yellow box. So this number indicate the number of patient case used for the model training in each cancer type. So the numbers are around 100 cases. That is very small size to train deep learning model. Based on my experience, minimum 300 samples are needed to train the deep learning model, but in this case, it is much less than that one. So I can say I tested my pipeline with these sparse data sets. And I used five-fold cross-validation method to train and test the model. This shows the example of the five data structure of five-fold cross-validation. The data set was split into training, validation, and test data. And by rotating the distribution of these three split data, it evenly use the whole data for training and validation and test. In the case of validation, this validation data set is used for early stop to utilize this early stopping method, and test data set is used to evaluate the performance of the model, and training data set was used to train the AI model. So in this case, because five data cases were used, five AI model was trained and five testing results are generated. So I used the average value of the testing results to compare to determine the other performance. And this shows the training process. It is a little bit complex, but if I explain it, in the case of whole slide images, I trained one time with the five-fold cross-validation. But in the case of transcriptome, because I automated the marker gene selection method, I tested a different number of macrogen sets. For example, I tested it with 100 macrogen, 200 macrogen, and to the 2000 macrogen set. So a total of 20 case was used to train transcriptome model. When I train the multimodal model, I use the combination between this single uh, whole slide image training case and each of the 20 um, transcriptome case. So as a result, the multimodal model also trained 20 times. There are 20 cases of the multimodal training. And this shows the architecture of the pipeline that I developed. So this shows the overall architecture. And this is the whole architecture has three the pipeline has three sub pipeline. One is the pipeline to train whole slide image model, and another is the pipeline to train the transcriptome model. And the third is the pipeline to train multimodal model. In the case of whole slide image model, the input data is like these whole slide images. And at the first step, tissue segmentation was performed. Tissue segmentation is that if we take the picture of the whole slide images, uh, there are real tissue part and the background slide glass part. So in this step, it automatically determines the real tissue part and just utilize only those images. And the next step is patch creation because, as I told you, the size of the whole slide image, file size of the whole slide image is almost 1 terabyte. So it is huge data, a huge picture. So that big picture cannot be analyzed all by once. So what this software does is it just split into small pieces of the images, and that is patch creation. And based on in each of the small pieces of the patches, feature extraction is performed using the pre-trained feature extraction model. Then the image data is converted into numerical features. And after that, this whole slide image model generate the split data, in this case, five-fold cross-validation. And based on this data set and the input feature, the model training is performed here and the evaluation of the model is also performed here and the result is reported. And in the case of transcription model, the input data is the TPM data, which is the normalized gene expression data. And in this module, uh, the TPM value are standardized and scaled, and also log2 transformed in this step. And after that, the marker gene selection is performed at this, and the split data set is retrieved from this whole slide image pipeline, and it also train and evaluate the performance of the model. And in this multimodal model, it utilized all the information which are generated from whole slide image model and transcriptome model, and also using the same split data. Um, it it concatenate the features generated from this whole slide image model and transcriptome model, and it train its own multimodal model, and also it reports the evaluation result. So originally, it is supposed to run each module by manually in the case of this CLAN pipeline. Then it is extremely complex work. So I um, put everything into a single pipeline. So now the users can run this pipeline with single command line execution. And also for high reproducibility, I containerized whole pipeline with Docker. So now the users can easily utilize this pipeline. And this shows the multimodal, how I perform the multimodal fusion by intermediate fusion. This left is the whole slide image model, and it is performed by Clam pipeline, and it used attention pipeline. And the last feature of the model is 512 features, and I used this for the multimodal fusion. And in the case of transcriptome feature, I used multi-layer perceptron, and the last feature is also 512 features. And this part is for the multimodal fusion. And I concatenated these two features and trained it with single-layer perceptron. So this is how I merged the two modalities. And this shows the tested model structure for hyperparameter setting. Actually, I tested a lot of different cases in transcriptome to determine the transcriptome model as multimodal fusion model. But in the case of transcriptome model, this multi-layer perceptron with one ski connection showed the best performance. And in the case of multimodal fusion model, I tested attention algorithm, attention with ski connection, and MLP with ski connection, but just simple multi-layer perceptron showed the best performance, so I used this. And this shows one example of the attention heat map of the ovarian cancer whole slide images. And here, this part is the cancer tissue, and this part is the stroma tissue, which is the normal healthy cell that include the immune-related cells. And the red and this blue color represent the attention score, and red is high attention score and blue is low attention score. This shows one case of one example of explainable AI model. Um, when train the AI model for whole slide image, this high attention area was used importantly. That means this normal cell was used very importantly to determine the therapy response. So through this attention heat map, we can determine which tissue is important to predict the therapy response of the patient. And this shows the development of the marker gene selection method. Actually, one big problem to develop the generalizable pipeline was the marker gene selection. The number of human functional genes was around 20,000 genes, but I cannot use that all 20,000 genes as an input to train the transcriptome model. So I had to pick the genes that are related to the therapy response. So I try to automatize it, and I tested a lot of different cases, but what I found is that the combination of the linear trend line analysis and Pearson correlation test worked best. So this shows the pipeline to automatize the marker gene selection, and it looks very complex, but just I made it as a automatic pipeline and integrated it into the master pipeline. And this shows the one example of the trend line analysis. And this dotted red line is the trend line, and each blue dot is the gene, and y-axis is a scaled TPM, the gene expression value, and x-axis is the sorted PFI, the sorted patient case by PFI. So as the absolute slope value of this trend line is higher, and the standard deviation of this gene expression is lower, it is considered as the high performance marker. So this shows the analysis result. Each dot represents the gene, and y-axis is standard deviation of the gene expression, and x-axis is the absolute slope value of the trend line. And these red dots are the genes selected as the candidate markers based on this method. And if you see the distribution, the red dots are distributed on the higher slope value and lower standard deviation. And this means this marker gene selection method works well. And also, the other problem was the optimization of training effort with the sparse data. If there is enough data, then I can easily use the early stopping method, which is generally used to train the AI model. Because the number of data set that I used was not enough, I couldn't this the performance of this early stopping was highly variable. It was not stable. So in this case, usually people use this fixed effort. But in my case, because I try to develop generally applicable training pipeline, um, this fixed effort could generate some method when the users use the different learning rate. So, to solve this issue, I developed a new method to use the elbow point of the training loss curve as the criterion to determine the training stopping effort. So usually training loss curve make this L shape of curve, and it you can easily find the elbow point of the training loss curve. Then we can determine the effort of this elbow point, and based on that, if we determine the training effort, training stopping effort, then even the users use the different learning rate that we can consistently find the training stopping effort. So I tested this method and by comparing it with early stopping method. And in this plot, the green triangle is the performance of the uh, model trained with this elbow stopping method. And as you can see, it consistently showed high performance in these four cancer types. So I determined it is working well, and I integrated this method in my pipeline. And also, I try to figure out if there is any pattern along along with the number of markers that I tested. So I tested from 100 to 2000 markers by 100 increment. I trained the transcriptor model based on this. So I try to find any proper or optimum number of the marker, but the result shows there is no pattern. So there is no deviation between the number of marker gene and performance of the model. And this shows the result. So I this shows the comparison of the performance of the models using the AUC value. Among the tested 20 different number of markers, I just chose the best performance model. And although the number looks very complex, you can just focus on the numbers highlighted with these gray boxes. So columns indicate the 10 different cancer type and rows indicate the modalities that I used. And if the best performance was identified from the multimodal model, then I highlighted it with these gray boxes. In overall, 8 out of the 10 cancer type showed very good performance. In the case of AUC value, if the AUC value is over zero point 0.7, we can say it is well-trained model. So 8 out of the 10 cancer type showed over zero point 0.7 AUC value, except only LUSC and OV. This LUSC is lung cancer and OV is ovarian cancer. And these two cancer types showed low performance, but 8 cancer types showed high performance. And one interesting thing is that not simply using as many modality doesn't necessarily train high performance model. So for example, CSC, LIHC, LUAD, and UCSC cancer showed high performance when it is trained with gene, microRNA, and whole slide images. But HNSC and PRAD showed the best performance with gene and whole slide image modalities, excluding the microRNA. But this breast cancer or the brain tumor microRNA and whole slide image multimodal showed the best performance. So this is kind of interesting result, and this shows the importance of the modality selection when we try to develop multimodal AI model. And this shows if the marker gene selection really works. So I used the I trained the model without marker gene selection method. So by excluding the low expression genes, this number of the genes were used in the training of the transcriptome model, but all the performance was much lower than the models trained with the selected marker. So I can say the marker gene selection truly works. Okay, this shows the conclusion. So, I could successfully develop the fully automated pipeline to develop multimodal AI model that can be applied to binary classification of the cancer clinical traits. And so the pipeline is applicable to various case types and high performance of and it can train high performance of multimodal models. And I developed a new marker gene selection method and I integrated it to this pipeline. And also for the sparse data set, I developed a new training stopping method. Okay, this is all I prepared today. Thank you so much.